Hi guys, and welcome to the last video of the Flip Learning lessons. Today I'm going to be looking at blood vessels and their function. So by the end of this video you should be able to describe the structures of the blood vessels and their functions. So we've got the arteries, the vein and the capillaries, and you're going to be looking at the arterioles as well a little bit. So what we have are the tough, fibrous, outer layer, the muscle layer, the elastic layer, the inner lining or the endothelium, the lumen. It resists the pressure changes that come from outside and inside of the vessel. The muscle layer is the part that contracts and controls the flow of blood. The elastic layer, and I think we've discussed this before in lesson before, is the part that maintains the blood pressure by stretching and recoiling. So such as within the aorta, we would have a large elastic layer. And then the inner lining is smooth to reduce the friction. And then the lumen isn't actually a layer, as I've drawn here, it's the bit that's inside. So the main differences between the blood vessels come about because of the varying proportions of each of these layers. So obviously we've got the lumen, which is the inner cavity, the lining layer, the elastic layer, the muscle layer, the tough outer layer. So these can vary in the arteries, the veins, and of course the capillaries to then allow them to carry out their function. How is the structure of the artery related to its function? So the main function of the artery is to transport blood under high pressures. So the sorts of adaptations that you would find in the arteries are a thick muscle layer, a thick elastic layer, the whole wall, so the actual all the layers put together is much thicker, and you will find no valves. So the muscle layer allows dilation or constriction in order to mediate how much blood can go through. So if we think homeostasis, so the thick elastic layer helps maintain the high pressures that you have from the heart going outwards through the arteries. So during systole, you will have a stretch, and then as they relax in diastole, they will recoil again. And the thick wall is there to keep the vessel intact. So it's a strong vessel because it needs to have high pressures, and therefore we can't afford to have breakages within the vessel. And then we have no valves because blood is flowing at such high pressures, there isn't a risk of it flowing backwards. So the muscle layer is relatively thin compared to the arteries. This is because the veins carry blood away from the tissues at a very low pressure. So constricting and dilating will not be able to control the flow of blood. So they will have a thin elastic layer in comparison to the arteries. This is because the blood here is flowing at a much lower pressure, so there is less risk of a burst of the vessel. So we've got a thin muscle layer, we've got a thin elastic layer. The thickness of the wall is quite small. This is again because there isn't a risk of any breakages. We're under low pressure, just trying to get back into the heart. Additionally, this characteristic of being quite thin allows the vessels to squeeze and flatten in order to sort of push the blood back up towards the heart. There are valves, and we know what the valves will do. So as you know, the capillaries are the site of exchange of oxygen, carbon dioxide and other nutrients in and out of the cells. What you will find is the lining layer, which is known as the endothelium, and it is one cell thick to allow the movement of nutrients and other gases. The lumen is quite thin as well. This is because the blood has branched off into all these various capillaries into very, very tiny spaces to then be transported into the cells. Obviously the lining, thin lining, is for rapid diffusion. Also, you would find that there are spaces between the cells in order to allow the cells such as the white blood cells to move in and out easily. Until now, we've been under the assumption that the capillaries are the final destination before all the metabolic components are able to pass into the cell to carry out respiration. But in fact, that's not completely true. What happens is that the capillaries can't actually get to all the cells directly. Therefore, we form something called tissue fluid, in which the cells literally bath in, and then we can reach all the cells like that. So tissue fluid is formed from blood plasma. What you will find, if I draw a capillary here, so the blood will flow, and in some cases it is able to carry out direct diffusion. However, most of the times the nutrients and all the required materials are able to form the fluid, which is this whole area here in which the cells can reside, and therefore all the diffusion in each cell can take place. Okay, so how does tissue fluid form? So basically the blood is pumped by the heart and passes down along the arteries. It then gets into the narrow regions, which are the arterioles, and then flows in to the capillaries. So there is a pressure that is obviously created by pumping from the heart. This is known as the hydrostatic pressure. So this is formed around this area. So this hydrostatic pressure that is builds up as you get towards the capillaries forces the tissue fluid to leave into the spaces in the cells, around the cells, sorry. So you have hydrostatic pressure here causing the tissue fluid to leave. However, here the hydrostatic pressure doesn't exist as much. This is because there is a higher pressure outside in the tissue fluid region in comparison to within the capillaries. Also, there is a lower water potential inside the capillaries than outside, this because of the plasma proteins that are within the capillaries. Therefore, rather than moving out, tissue fluid will now move back in. So the idea is that the hydrostatic pressure within the capillaries is only maintained up to this point, so that all the required tissue fluid can leave and then reach the cells. But then at this point, the pressure is no longer there, so that tissue fluid does not continue to leave. So in this model, which is called ultrafiltration, only the very small molecules will be able to leave. So the larger molecules aren't able to leave, such as the plasma proteins and the cells of the blood. Like I said before, once the cells have used up the metabolic materials that they need, the tissue fluid can now flow back into the capillaries and go back to the heart through the vein. So how does this happen? As discussed before, the hydrostatic pressure reduces greatly as the tissue fluid leaves, so the tissue fluid will want to flow back in and go back. So we would have a higher hydrostatic pressure outside in the tissues in comparison to inside, so the tissue fluid would like to flow back in. What has also happened in this situation is the water has moved out, in this case, leaving the plasma with a lower water potential. So Therefore, the higher water potential in here, water, will force the tissue fluid back in as well. As well as this, the tissue fluid has lost oxygen and other nutrients to the cells, but when we're going back, it has picked up waste materials and CO2 to take back. So this system is quite effective in forming tissue fluid within to bathe the cells, and also to pick up the tissue fluid back into the plasma to then go back to the heart. However, not all of the tissue fluid can return in this way. So what happens is the remaining tissue fluid can enter through the open ends of the lymphatic system, which then go back into larger vessels and join in veins near the heart to then drain back into the blood. So the lymphatic system carries the tissue fluid, which is now termed lymph. It doesn't receive pressures through a pump like the heart. Therefore, it uses hydrostatic pressures, which we've looked at, and it uses contractions of the muscles that surround it. So this allows it to flow around the body. So if you have a look at figure 7 on page 182, it gives a nice summary of how tissue fluid forms and returns in the bloodstream. So what you have is the tissue fluid, which is formed in the cells. It drains into the lymph, it returns to the blood plasma through the veins, and then of course we've seen how in the capillaries the tissue fluid can leave. It can be picked up towards the venous end of the capillaries to then return to the heart. So that was the end of the video, guys. Hope you enjoyed it. Catch you later.